Gives me great pleasure to introduce our speaker here this morning, Dr. Kenneth White, certainly one of the most outstanding of our many outstanding faculty members and researchers at Utah State University. You're in for a real treat this morning as we see some of the exciting work that Dr. White has been doing with animal cloning, but what is particularly of interest to me is the application of that research to human health and human pregnancy, and so I'm excited that he's here with us. Dr. White is department head and professor in animal, dairy, and veterinary sciences at USU. He's also currently interim director of our Center for Integrated Biosystems, and importantly is one of our lead scientists in the recently created Veterinary Diagnostic and Infectious Disease USTAR program at Utah State. And so for those legislators who are here, thank you for USTAR. And one of the things that Ken will do is allow you to see some of the exciting research that is coming out of USTAR. Ken, thank you and welcome.
Thanks, President. Thank you, President. I'll just echo something that President Ulbrich said. You know, it's a real honor to be here to be able to speak to you this morning. But, you know, keep in mind that I'm just one. You know, we'll talk about one program, but there's a whole laundry list of exciting programs at Utah State University, wonderful things going on there, and it's been really my opportunity and my honor to be a faculty member at that institution for the past almost 19 years. It's a wonderful place to work. We have excellent students, and I just couldn't ask for a better atmosphere and a better support from our administrators and from the institution, the university as a whole. I've got a couple of friends here. I appreciate, and they're probably the only two I have. I saw Dr. Ross Smart. Ross was a longtime faculty member in the ADVS department, and a tremendous asset, not only for ADVS and Utah State University, but also the efforts of the state of Utah in the area of diagnostics. And so it's just a real treat and pleasure to have Ross here. I also noticed Dr. Olson. I had the opportunity to meet him, I guess, about a year ago, and listen to some of the interesting and awesome accomplishments that he made in his career and continues to make today. And so, again, it's just my honor and pleasure to be here. This is obviously a topic that's very near and dear to my heart. It's a topic where Utah State has been able to generate a tremendous amount of interest, both nationally and internationally. We are considered one of the leaders in this area of research in this country, certainly, and in the world. I'll try and point some of that out as I go along. In 2003, when we were able to, fortunately, to partner with University of Idaho and produce the world's first equestrian clones. That work was on the front page of nearly every major newspaper in this country and several outside of this country. One illustration of the national recognition that Utah State has received is that, and I never thought I would ever be excited about being associated with the term geek, okay? But I actually was. In the fall of 2008, Popular Science ranked the 10 Geeks Guide to Colleges, the 10 smartest universities in the country, and Utah State, of course, is listed there, along with some pretty prestigious Stanford, Berkeley, Wisconsin, Cornell, Boston College, and again, it was kind of an acknowledgement of the cloning work and the opportunity that we provide for students. Very unique opportunity, particularly because we use nothing but domestic animals and primarily cattle in our research. And I'd say we're probably the only institution in this country that has the ability to do that. And there's probably about 6 in the country that actually do much cattle, nuclear transfer. But again, we're pretty proud to be one that kind of leads the pack. What I'm going to do is touch on 3 pretty basic topics today. First is I want to explain a little bit about the process of somatic cell nuclear transfer so you can understand a little better, and then illustrate just very briefly some of the application of why this is important for agriculture. Then I'll suggest that and provide you a little bit of information to show it makes a very good model for studying inappropriate gene expression. And then I'll talk lastly about a topic referred to as epigenetic. They provide an excellent epigenetic model, and I'll explain what epigenetics is. Essentially, in a few slides from now, but essentially it's the part of traits and gene expression that isn't affected by the sequence, but more affected by the things that bind in and around specific sequences. So cloning is not something that's new in agriculture. When I was at the University of California in graduate school in the early 80s, we were cloning animals. We used a much different approach. We broke off a little sliver of a double-edged razor blade, used a little super glue to glue to a micro pipette, and we had a microsurgical blade. And we'd use that blade to mechanically cut a pre-implantation embryo in half. We could remove half of that embryo, transfer it to an empty shell, and thereby create two identical twin embryos. Those could be transferred to surrogate moms, and very high success rate. Excuse me. And this is an example of two of those identical twins that were produced. And again, cloning helped us understand a little bit about development that we kind of recognize now, in that they don't always look exactly the same. When you have animals that have coat colors, some of these early experiments helped us understand that actually there's two components of the pigment cells. Okay? One is genetically controlled. In other words, how far down the backbone those cells migrate, or those progenitor cells migrate. But then they migrate from the backbone down to the stomach. And that part of it's random. So they'll have colorations that are generally in the same areas, but they won't look exactly the same. Now, from that point, we move to the type of cloning that we do nowadays, where we use an unfertilized egg. There's a magical, there's magic associated with unfertilized eggs that we don't really understand what that magic is. But what we can do is we can, and the DNA in this example has been stained blue, okay? And you can see part of it here and part of it there. We go into these unfertilized eggs and we remove all the DNA, all the nuclear components of that egg. So what's left behind is a membrane and then the cytoplasm, the stuff that's not DNA. Now, you'll maybe remember, for those of you that had beginning biology, that we get half of our DNA from mom, half of our DNA from dad. The eggs have a magical, kind of an interesting way of getting down to that half DNA. They take the half they don't need, and they create this little structure. It's called a polar body, and it extrudes the extra DNA they don't need. So essentially what we're doing is removing that polar body. Now that polar body also is a nice landmark for where the remaining DNA is inside the cytoplasm of the egg. So we also remove that at the same time. So that, and this is just a series of slides that kind of shows you that process. So again, if you look at about 4 o'clock, you can see that polar body. We take the pipette, we remove the polar body as well as some of the adjacent cytoplasm. Go down, there's a herd of cattle here. We pick up one particular donor cell that contains the DNA. We bring it back, go back through the same hole, place it here adjacent to this egg cytoplasm, okay? And that's, the process is as simple as that. Now what I'd like to do, this is what you'd see if you were actually looking at through the microscope. And again, you'll see that first polar body, and then inside the egg is the remnant of the DNA that's going to, if it was fertilized, would combine with the sperm's DNA. So we go through the shell. And of course, I assume everybody knows this is pretty small. So you saw the polar body go. Now you can see the cytoplasm stretching away, and we're pulling out the DNA that's inside the egg. And then what you'll see is we'll stretch that out. The membrane will pinch off. We'll remove it from the egg. The membrane will pinch off, and then we'll discard that unwanted DNA. And again, the egg's just held by suction on this little holding pipette. Now this will go out of focus because I need to go down the bottom of the dish. I have these donor cells. And we'll look through the pile of donor cells. We'll find a nice small round one. Pick that up, and then we'll do the transfer. Okay, and you see that donor cell? We aspirate it into the pipette. Now we'll go back, and the egg will get back in focus. We go back through the same hole. And we transfer that cell and make sure it's kind of tightly packed against the egg. And you can see the donor cell right here at about, 1 o'clock. So, and it's as easy as that. Now, in relative sense. So now, you might have picked up that nucleus from that donor cell isn't inside the egg. In order to accomplish this, we take that, what's referred to as the couplet of two cells, put it between electrodes, and we use electricity. We pass a current through that. Now, because of the chemical properties in the membranes, that electricity disrupts those membranes. Then they reform in the stable state that they prefer, and rather than reforming as two distinct membranes, they reform as a single membrane. And in that process, we introduce the nucleus from the donor cell into that cytoplasm of the egg. And again, you can see, hopefully you can see that fusion process has started to occur in this example. Now, the birth of Dolly was a hallmark from the standpoint of nuclear transfer research, because those experiments showed that we could take a differentiated cell from an adult animal. It could be repackaged, reprogrammed, and actually produce a live offspring, which was a remarkable concept. Folks that worked in plants and some amphibians knew that could occur, but we'd never been successful in doing that with mammalian cells. That work was actually preceded by work by the same laboratory where they used a somatic cell that was from an embryonic cell line. So again, you can get 3 different types of somatic cell. Somatic cell just means a cell that's been differentiated, okay? So you can get those from embryo cultures, fetal cultures, as well as adult cultures, okay? Now, since Dolly, there have been about 20 species that have been successfully cloned by use of the somatic cell nuclear transfer. I haven't listed all of them. Here's a few of them. I'd like to draw your attention to pigs. Utah State has a little bit of a tie to the pig work. And in fact, the scientist that did that work, Irina Polozheva, was up on campus for the past 2 days and still up there, and I'll join her when I go back. She was actually a postdoc in my laboratory from 1993 to 96, and she now is the chief scientific officer for Viagen, which is a cloning company located in Texas. But they were the first to produce live pigs using somatic cell nuclear transfer. Of course, our work was the first to produce an equine species when we announced the birth of 3 identical clone mules. And that'll be another talk. That was an interesting, to take a species that can't reproduce and actually reproduce them was an interesting feat. Now, let's talk a little bit about duplicating animals with superior traits. That's where somatic cell nuclear transfer really has some utility as a tool for assisted reproduction in agricultural animals. So you find animals with very, very outlying traits that are unique and that are economically important. You could identify animals that have a lower carbon footprint. There was a report about 3 years ago of a dairy cow that was extremely high in omega-3 fatty acid, lower in some of the detrimental fats. So that would be an animal. I've given you a couple of examples that should hit home really easily. High milk production, superior carcass traits. And I'll spend just a little bit of time explaining carcass traits. Understand there are 2 things that are problematic with selecting carcass traits, okay? Number 1 is that the animal is dead, okay? So when we know that this is a carcass that is exceptional, the animal's already been dead for 24 to 48 hours. I taught reproduction for many, many years. That's a real problem for reproduction, okay? The second thing is, that's equally as difficult a problem, is that the male animals are castrated when they're very young animals as a management practice so that they'll grow better and more effectively produce the type of carcasses we're interested in. Again, I go back to my reproductive textbooks. That's also a very critical problem. So, and again, for any that don't understand what I'm talking about when I say carcass traits, I've given a couple of examples of two of the carcass scores that we commonly see associated with these animals. One is the quality score, and what you'll notice hopefully is that I've put various different grades of meat, and there's commercial down here, but what you'll note is the level of marbling differs between all of these. Well, marbling is very important for a piece of meat. If you want a flavorful, there's probably a higher probability it's going to be tender, and it's actually going to be a far more expensive cut of meat to be a prime. Now, the second grade are the yield grades. And what I draw your attention to is this white line. This, number 1 being the best yield score, number 5, the poorest. And you'll note that how much more fat there is here than what you see on the number 1. Now, most of the consumers, if I polled in this room, if they saw the two of these pieces of meat in the supermarket, would all gravitate towards this one. Butchers know that, so what they end up doing is they cut this off. Well, when you've paid, you bought your meat a certain price per pound, this piece of meat just got more expensive to produce, okay? So the best carcass you can have is a prime yield grade 1. Now, you'll notice that anybody who's ever shopped for meat, you never find prime, right? Choice. The reason why you don't is because less than 0.5% of the carcasses that are collected each year will grade out in prime, and even far less that will grade out as prime yield grade 1. So what we've done is to take tissue from some of these animals. Once we know that they're prime yield grade 1, this is an example of an animal that's a prime yield grade 2. We've produced approximately 60 animals. Some are from the same cell lines, some are from different cell lines. We're using those animals as seed stock animals to breed in a commercial herd to see whether or not we can increase the average quality carcass scores by using these animals as breeding animals. Now, prime beef has a premium price tag. If you have a higher quality prime carcass, you'll get $150 more than your competition. So there's a huge economic incentive to be able to increase the quality scores of our carcasses. Now, pregnancy complications. There are complications that are unique to nuclear transfer pregnancies. And the biggest being the high rates of pregnancy loss throughout gestation. This is work by another lab, retrospective studies, where they went and looked in the literature to evaluate what the pregnancy rates were. What we find, the green bars represents the control pregnancies, so the embryos are all transferred, that's why it's 100%, and then 21, 35, or 50, 70, and so forth, days of gestation until they deliver. What you'll note is in all of these, when their embryos are transferred, you get an immediate drop, but then pretty much the pregnancy rate stabilizes from about 40 days gestation until term. Now, regardless of what type of somatic cell you use, whether it's an adult, fetal, or embryonic, we see that we get a similar initial drop, but then throughout pregnancy, those pregnancies continue to be lost, okay? The reason why we believe that those pregnancies are lost is because we've found that the placenta that forms in these animals is aberrant, or it's not normal, okay? I've got an illustration, and in the next slide I'll show a quick real life so you'll believe me exactly what they look like. But what we see, this type of placentation in cattle is what we refer to as a cotyledonary placentation. So the fetus has a cotyledon. Its counterpart on the mom's side is referred to as a caruncle. They come together. That's where exchanges are made between the fetus and the mom, and that combined structure is referred to a placentome. What we see in a normal placenta is evenly distributed cotyledons. They're uniform size, they're a healthy kind of pink color, all suggesting a very wholesome and healthy environment. If we compare those to what we find in cloned placentas, we see they're far reduced, they're much more sporadic with where they're located, they're far larger, almost as a compensatory response, and we see that the color is often a very stressed color. And the next slide gives you an example of 120-day normal placenta from cattle pregnancy. And again, you notice these cotyledons are pretty well distributed, nice pink color. Contrast that to what we find in a similar 120-day placenta from one of these cloned animals. So I mentioned epigenetics, and let me quickly try, and this is Utah State is known for economic education, okay? This is going to be your economic education on epigenetics, okay? What we find is that when these differentiated cell nucleuses go into that egg, remember I said it's got magic components. Those components have the ability to remove these epigenetic markers from the DNA, so that you take a cell that only knows, say a skin cell, that only knows the genes that are supposed to be expressed in skin cells, and conversely, all the genes that are supposed to be shut down in skin cells, you put that in that environment of the egg, and those markers come off so that that nucleus can become any type of cell in the body. And that's requisite for being able to have normal development. And that methyl groups, acetylation points, those are all the things that kind of tag onto these DNA that either excuse me, target genes to be expressed or target genes to be shut down. Okay? What we think happens is that when we do nuclear transfer, the nuclear reprogramming is often incomplete. Okay? So we see and we anticipate differences in gene expression. Okay, so file that away. I'm going to give you another quick lesson, okay? You're really getting your money's worth here. So I'm going to talk to you about how gene expression works, okay? If you can think of DNA as the blueprint. Blueprint's very valuable, but if any of you that have ever built a building or constructed a house, you know that the plumber needs to get a copy because there's some specific things for the plumber. The electrician needs a different copy. Framing guys. So, but we can't let these leave. These blueprints, DNA cannot leave the nucleus. So, the way we use a Xerox machine to make copies of those blueprints so that we can give the subs their copy, and then they can do whatever they need to do, and you get the house built. Our Xerox copy is referred to as mRNA, or mRNA, messenger RNA transcription, and I've designated the messenger RNA, or the gene expression, as these red little squiggly lines. Now, they can leave the nucleus, and they have to leave the nucleus. Once they leave the nucleus, those, just like the electrician takes those copy of the plans and turns them into an actual wiring system for the building, that messenger RNA is translated into a finished product or functional protein. Okay? Now, we use these messages and we detect those by the use of what's referred to as a gene chip. So we can take a control embryo, we can take a cloned embryo, we can take the somatic cell that made the cloned embryo, and we look at the profile of genes that are being expressed with this gene chip. When we do that, we did one set of experiments where we took cloned embryos and normal embryos, what I call normal or control embryos, and we took tissue from the cotyledons, and we looked at what genes were being expressed, and we compared those, and we found that there were several genes that were differentially expressed in the cloned embryos as compared to the control embryos. We also found several genes that were differentially expressed when we looked at the placental tissue of normal animals compared to clone animals. Interestingly enough, what we found was that there was one gene that was common to both sets of data, only one. And it's listed here, it's this MHC gene. Now, MHC proteins are the proteins that help your body distinguish self from foreign, okay? So MHC proteins will elicit a rejection response, okay? Now, this just illustrates the differences in expression. So what we saw with the clone tissue versus normal tissue, and you can see some of those differences are pretty severe. Others are very subtle, okay? If we compare these boxes to these 3 boxes. Now, the MHC, I have a colleague that's at Utah State now, and before he was at Utah State, Chris Davies, he did some experiments independent of ours. So we ran these genes and we found this MHC protein was out of whack, okay? He did some experiments and he actually looked at the cells. Now, this is where the MHC story gets pretty interesting. So if we look at control pregnancies, what you're looking at is microscopically, you're looking at the mom, that's the interface between the mom and the baby, okay? The interface right here, mom, baby, okay? And what you'll find is he's stained to try and detect these MHC proteins. And what you see in this control, and if they're there, you'll see this red color. So he picks up red color in the mom's side, again here, and a little bit down here. But if you look at the fetal side, there's nothing, okay? If you compare that to the clone pregnancies, the same tissues in the same areas, you find a tremendous amount of red color. Again, the interface being right here, you find a tremendous amount of this red color, which means MHC protein being expressed, okay? So what we think is happening is that you're getting, the clone is being rejected, the placenta is being rejected, okay? So the important conclusions from this were that we had that one single gene, an MHC gene, that was aberrantly expressed in both types of tissue that we looked at. One was the embryo, the other was in the placental tissue that embryo generated. We found that because of this data and combining Chris's work with our work, that clone fetuses serve as an excellent model to study human placental insufficiency. You have a fairly good percentage of human pregnancies that are lost because the same type of a situation exists and an immune response is generated at a time when it's not supposed to be generated and the pregnancy is lost. So we felt like by using cloned fetuses, cloned pregnancies, it would increase our understanding of the communication between a fetus and the mother's immune system, okay? Well, we were able to convince an NIH review panel of that same argument, and we were awarded a major NIH grant, and we're currently in the first year, actually starting the second year. And we hope to understand MHC expression in these cloned fetuses during pregnancy much, much better. And hopefully determine if the immune-mediated abortion can be avoided by, in some way, masking or removing that MHC trigger. So hopefully it'll be a big help to the condition in humans, but at the same time, hopefully by the work and the research we do, we'll be able to understand why pregnancies are lost in cattle as well, and hopefully be able to increase our efficiency of that process from where it is at about 10% today to hopefully about 40 to 50%. Now, President Albrecht mentioned the U-STAR program, and we were very happy to become a part of that. And this funding is going to do a couple of things. The main focus for this U-STAR program, Veterinary Diagnostics and Infectious Disease, or VDID, as we prefer to refer to it as, we want to develop next-generation diagnostic assays. Currently, some of the accepted regulatory assays that we use to kind of surveillance purposes for our animals, as well as for diseases that are important for animals, but also diseases that could have impacts on the human population. A lot of those were ELISAs, a lot of them were developed, some of them in the 80s. And so the goal of this is to develop more molecular tests that will increase the sensitivity and also increase the specificity and the selectivity, so we can more carefully distinguish different types of viruses, some of which are very concerning, others which are less concerning. Now, cloning, how does cloning tie into this? Well, one small component of it is animal models. So keep in mind, there are many diseases where it's difficult to study not only the diagnostic component, but also the therapeutic component, because there's not a good animal model. Okay? So one example of work that we've got going right now is where we take somatic cells and we mutate the genes in those cells, and in this case, to have the gene sequence that will make these susceptible to human cystic fibrosis. Okay? We make these, when we make these somatic cells, and we're going to use the sheep as the model, we can then take those somatic cells, clone with those somatic cells, and produce animals that have that specific defect that actually allow them to develop cystic fibrosis, so then we can look at therapeutics and other things to help lessen. Many of the folks with cystic fibrosis end up dying of pneumonia. And so it'll provide valuable models for diseases such as cystic fibrosis, as well as other infectious disease models that will allow us to develop not only diagnostics, but hopefully better understand some of the mechanisms of the disease. So we are very grateful for the U-STAR funding. Reprogramming Dynamics. I threw this in just so that you'll know that all three of these mules are healthy. Some of the guys got a little bigger bellies today, but the mules are doing quite well. That was actually the day of the breakout race. They were 3-year-olds. It was in the hotbed of Winnemucca, Nevada, okay? And they ran races, and actually this one won, and that one got second place, and this guy was just a show boy. Now, remember I mentioned how different gene expression is in a somatic cell versus an embryonic cell. And this is an example. You can see we've selected 5 genes, okay? And each of these columns are just replications, okay? We use the somatic cell. But you can see if you compare the gene expression of this particular gene to embryos, you can see that it's dramatically different in all of these genes that we looked at. So what we wanted to do is, we know that gene expression is different, but what we didn't know is how quickly and how much fidelity there is when the nucleus goes into this magical egg at making that nucleus look more, and the gene expression profile look more like an embryo's, okay? So again, we chose these where there was big disparity in the expression patterns, and I'm going to I'm not going to spend a whole lot of time on this, okay? So don't go to sleep. So the green bar, okay? 3 of these genes have very high gene expression profiles in the differentiated or the somatic cell. This little tiny bar is what the oocyte looks like before the nuclear transfer. The yellow bar is our nuclear transfer embryos at 30 minutes, okay? So that's 30 minutes after the nucleus has gone into the egg. The gene expression profile for this particular gene goes from there to there. This is 12 hours, 24, about 3 days, 4 days, 5, and 7 days, okay? And what we were trying to illustrate was how dynamic these changes were. And what you can see is that in some examples, it's not until 7 days before the gene profile in the cloned embryo looks like the control embryo. This OCT4 is a very important marker for stem cells and pluripotent plasticity, okay? They can become any kind of cells, undifferentiated, kind of an undifferentiation marker. The interesting thing is that you see in these control embryos, the expression levels are really high, okay? They drop at this point because actually in development, differentiation starts at the morula stage. So by this 24 hours, differentiation has already occurred, but the interesting thing to us was that the difference between the clones and the control embryos throughout that whole developmental period. So again, here are some examples. These three are examples where the somatic cell has no, and again, we would expect no gene expression from OCT4. I just told you that's an undifferentiated. It's associated with undifferentiated cells. Okay? So in fact, we don't. But if we look at this, we see lower gene expression of this very important gene throughout development. Okay? Who cares? Okay? I mean, you know, probably about 10 minutes, maybe 15 minutes ago, you were asking yourself, who cares? Well, the results, if you think about it, the results from these experiments are critically important if we want to develop stem cell applications. I mean, I'd love to be able to generate these, what you all hear in the media, embryonic stem cells. I'd love to be able to generate them without embryos. Now, they're induced pluripotent cells where they take 4 genes, one of them's OCT4, and they make these cells, and they call them stem cells. And to a certain degree, they're stem cells, okay? It's kind of like I've got a grandson that's got a Peyton Manning jersey on, and he looks like a football player, but he's not a Peyton Manning, okay? Kind of the same thing with these induced pluripotent stem cells. They're a stem cell because they're wearing the jersey, but they're really not a real stem cell, okay? So by understanding how these genes are turned off and turned on, the hope would be that someday we could take a differentiated cell, you could go into your physician, take a small biopsy of your skin, the lab rat could then take it back in the back room, throw it in a dish, do some things that we learned how to do through this process, and turn it into a bona fide, genuine stem cell that could then be rushed back in the room in a few days, injected in you. It could circulate and get located, targeted back to, say, your liver that's failing, and repopulate that liver. It's from your own cells, so you don't have a rejection response. So you have, hopefully, a healthy liver, okay? It also is important for clinical discoveries, for disease prevention and therapies, okay? Many cancers are because of the misregulation of genes that are very, very important in early development, but are genes that you don't want on later on in life, okay? So being able to solve some of these mysteries and be able to figure out how genes are regulated and the dynamics of gene expression is critically important in these areas. Future. I'm going to guess the future's probably not going to be this, okay? But understand, somatic cell nuclear transfer is probably going to be one of the tools in the toolbox, okay? Certainly in agriculture, from an agriculture standpoint, it's going to be one of the tools in the toolbox to be able to reproduce animals for assisted reproductive purposes. I'll suggest that it's also going to be a valuable, and has become an extremely valuable tool for understanding much better some human diseases that are critically important for us being able to solve the mystery of how they develop and how to therapeutically handle them. I just wanted to provide acknowledgments. Remember I told you my geek guide slide? This slide represents all the students that I have had or currently have working on this project. This list are graduate students. Kai Aston received his PhD about a year or so ago and is down now at the University of Utah on a postdoctoral fellowship. The remainder of these are graduate students. All of these are undergraduate students. A couple of them, for example, these two were actually on posters on the Hill just a few years ago. Mitch is now in medical school in Duke University, and John is now in veterinary school at Texas A&M University. So these are phenomenal kids. They've gone on and will go on to do many great and wonderful things. I've got a research associate and a postdoctoral fellow, both from China. J.R. Simplot Company has been instrumental in allowing us to do much of the live animal work and the transfers and providing, and are doing now the breeding studies to evaluate whether these breeding animals that were selected by and produced by nuclear transfer for the carcass traits can actually increase the quality of the carcasses. And then, of course, I mentioned Dr. Davies is critical to our work. He and I are working together on our NIH proposal, as well as Dr. Bunch and Dr. Rickards. So again, a lot of, and oh, don't let me forget Dr. Stevens. Woo. When you're looking at all these numbers for all these gene expression experiments, trust me, you want a mathematician and a statist and packaged in the same body to help you out, to help figure out what you're supposed to be worried about and what you're not. So with that, I'd be happy to answer any questions you might have.
California, the cells that you're cloning, where do they mature? Do they have to go back into a surrogate animal, or?
Okay, yeah. So let me take it from the video. Okay, we do that, the cloning process. Okay, and we fuse those cells, and then we go back into the laboratory. Well, we're still in the laboratory. We put them in an incubator, and we culture them in the test tube for 7 days. After that 7-day period, they're at a stage that is very convenient to do a non-surgical transfer to a surrogate female cow, animal. And so we transfer those embryos to the surrogate mom, and then it's about a 280-day gestation period, subtracting the 7 days that they were in the test tube, and they're born. I mean, that's one of the reasons why, I used to have flowing hair all over my head. When you're having all these losses during pregnancy, you've got to wait 9 months. You got to be pretty thick-skinned and able to take disappointment, because there's a lot of it. Go ahead. Yeah, I think you have a follow-up.
Can any female animal be used as a surrogate? Or
So let me give you a little.
They don't have to have already produced offspring to be
No, and they just have to be cycling. Okay, so they have to be old enough where they're cycling, and I think one of the reasons why I'm so passionate about having undergraduates come into my laboratory and work is because I remember when I was an undergraduate and a junior, and I had a small family. Junior probably, or senior, I don't remember. Anyway, I had a lecture by one of my faculty who talked about embryo transfer. Okay, now this was like in '78. Embryo transfer is a big deal in cattle. It's like no big deal now, but then it was like cutting edge, and that's kind of what I thought was really cool. So, and again, this principle's the same. You take a very expensive or an elite animal, and you can use a protocol that will allow her to generate, instead of 1 embryo, maybe 6 or 8 embryos, and then you take and remove those embryos by just flushing them out of her, and you can take and put them in other cattle, other recipient females, and allow them to carry those embryos to term. Okay, so long as they're cycling, old enough to cycle, they can be used as a recipient animal. Yes, sir.
So once the carcass has been improved and you've achieved the quality of meat you want, does it go back to a normal sexual reproduction cycle, or is it always caught in a cloning process?
Let me try and answer that. I'm not sure. So when we clone these animals, again, it's a tremendous amount of work to do that. So what usually happens is that these animals are used as breeder animals, okay? So they breed and they produce offspring. Now those offspring are the ones that we're evaluating for lowest quality. So those cloned animals we produce breed with control, if you will, females, and they produce what we call F1, or the offspring from those. Those offspring are the animals that we're evaluating the carcass traits. Now, heritability on carcass traits is anywhere from about upwards of about 40%. So there's probably a very high likelihood that if we selected these animals and they have the genetics that equate to a high quality carcass, those traits are going to be passed on. And some of the data we're seeing is supportive of that.
You, looking down the road, foresee the ability to accelerate this gestation cycle at all? I mean, it seems like that's one of the constraints here.
Yeah, I think I don't know that we can I mean, it takes a certain amount of time, 9 months to be able to do this. It's probably it's beyond my gray matter to see a scenario where we can speed that up. However, we can speed up some of the generation intervals because cloning, you can, rather than having an animal that's sexually mature, you could actually take cells from a very young animal, take those in the lab, and actually start producing offspring from her in a much shorter period of time. Okay? You're welcome.
I want to thank Dr. White for his presentation, and he'll be around to answer some questions if you've got individual things that you'd like to ask. So we're grateful for your time, Dr. White.
Absolutely.
Thanks very much.
Thank you.